Hello everyone, my name is Nana and I'm from the Bodhi Academy. So in our last video we have seen about the glucose, structure of glucose, both straight chain and cyclic structure. Now in today's video we will see about the fructose. Now glucose and fructose both are the hydrolytic products of sucrose. So fructose, it is an important ketohexose. Now what is glucose? Glucose is an aldohexose, whereas fructose is a ketohexose. Now it is obtained along with glucose by the hydrolysis of a disaccharide, that is sucrose. As I already told you, it is a product of hydrolysis of sucrose. Along with glucose we get fructose. Glucose is a aldohexose, whereas fructose is a ketohexose. Now it is a natural monosaccharide found in fruits, honey and vegetables. So it is a natural thing, natural sugar which is found on fruits, honey and vegetables. Now it is pure, now its pure form, it is used as sweetener. Okay. Now when it is present in its pure form, we use it as a sweetener. Now it is also important as an ketohexose. Now, what is the structure of fructose? Now, fructose also has a molecular formula of C6H12O6. Just like glucose, the molecular formula for fructose is C6H12O6, and on the basis of its reaction, it was found to contain a ketonic functional group, and at carbon number 2 and 6 carbons in straight chain, as in case of glucose. Now for glucose, I have shown you the reactions to prove the presence of an aldehyde group, right? How we have proved the presence of aldehyde group? By oxidizing it with mild oxidizing agent, that is bromine water, right? Now here, by doing some basic reactions, it was found that there is a presence of ketonic functional group, and this ketonic functional group is present at carbon number 2. It's present on the second carbon, and again same thing, that the 6 carbon atoms are present in a straight chain structure. Now it belongs to a D series. Again, this also belongs to a D series, and here it is levorotatory. For glucose it was dextrorotatory, right? Now for fructose it is a levorotatory. Now it is approximately written as D minus fructose. For glucose it is D plus glucose, right? So here it is D minus fructose. Now its open chain structure is shown here. Now this is the open chain structure of fructose. Let's write the open chain structure of fructose. So we have again 6 carbons. 1, 2, 3, 4, 5 and 6. Now on first carbon, on the second carbon we have a ketonic group, right? By conducting various reactions we have found that there is a ketonic functional group at second carbon. So on second carbon there is a ketonic functional group. Now first carbon, again we will have a primary alcohol. Okay. Now H, OH. So as usual, third carbon, the OH will be present on the left hand side. Okay. OH, H, OH, H, and as usual you will have a primary alcohol at the bottom also. Okay. Now this is the straight chain structure of fructose. Now it also exists in 2 cyclic forms, which are obtained D minus fructose by the addition of OH at C5 to the carbon ketonic group. Now just like glucose, fructose also has 2 cyclic forms. Now in glucose, the fifth, here also same thing, the OH from the fifth group is involved in the formation of a cyclic structure. Okay, so here the OH from the fifth carbon will bind to the ketonic group. Okay, will bind to the ketonic group. There the OH from the fifth carbon was binding to the aldehyde group. Here it will bind to a ketonic group. Now this ring thus formed is a 5-membered ring and is named as furanose with analogy to the compound furan. So there it was with 6-membered ring, right? Here it is a 5-membered ring and is named as furanose, whereas glucose it is pyranose, fructose it is furanose, and this furanose is from the compound that is furan. So this is the furan. Okay, 5-membered ring with a oxygen. Okay, so furan is a 5-membered cyclic compound with 1 oxygen and 4 carbon. Okay, 1 oxygen and 4 carbon atoms. So 1 oxygen and 4 carbon atoms. Now alpha D fructofuranose, how you will form? The 5th carbon, the OH, this will go and bind to the 2nd carbon, because here we have 2nd carbon with ketonic group, right? So here the O from the, I'll again show you the structure. Let's draw a fructose structure. Two, three, four, five, six. 6 carbons. Now first carbon will have a primary alcohol, that is CH2OH. Second carbon has a ketonic group. Third carbon will have as usual OH and H. So this is the straight chain structure of fructose. Now here what will happen? From the 5th carbon, okay, from the 5th carbon, this O is the one which is connecting both the structures and creating a cyclic structure, right? Now how we will draw a cyclic structure? So 1, 2, 3, 4, 5. I'll draw only 5. Okay, I'll show why I'll draw 5. Now this is carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. Now where did the 6th carbon go? Okay, so since you can see this oxygen, okay, so here it is like this. Okay, so this will be CH2OH. Here you will have H. Here OH, H, H, OH. Now the OH from this, okay, the H from the OH group of 5th carbon, it will bind with the oxygen of the ketonic group. So here you will have a OH and this CH2OH will come here. So CH2OH. So this is their 6th carbon. So this is the cyclic structure of fructose. Okay. So this is the cyclic structure of fructose that is formed. Now so here also you can see the same thing that I have drawn, right? So 5th, the first carbon has come down, right? So here also you will have a alpha and a beta structure, because alpha means the OH will be on right hand side, beta means the OH will be on left hand side. Now these are the cyclic structure of fructose, or the 2 anomers of fructose. So when we draw a furan structure and placing the molecules appropriately within them, just how we have drawn for glucose, similarly when we draw it for a fructose. So this is a fructofuranose, and that is alpha D fructofuranose and beta D fructofuranose. Okay, these are the examples of monosaccharides. Now let's move on for disaccharides. Okay, so monosaccharides we have seen glucose and fructose. Now disaccharides, you have already read that disaccharides on hydrolysis with dilute acids or enzymes yield two molecules of either the same or different monosaccharides. This was the basic definition that we have given for the disaccharides, that when we hydrolyze a disaccharide with dilute acid or an enzyme, what we get is two molecules of monosaccharides, whether it will be a same or a different. Okay, like glucose glucose or glucose fructose. Now, the two monosaccharides are joined together by an oxide linkage formed by the loss of water molecule. Okay, now since sucrose is a basic example of a disaccharide which is formed out of glucose and fructose, which are monosaccharides. Okay, these are two different monosaccharides. By bonding, there is a bond between these two monosaccharides to form a sucrose, right? The two monosaccharides are joined together by an oxide linkage formed by loss of water molecule. Now these two monosaccharides are joined together by an oxide linkage. Okay, there is an oxide linkage between these two glucose and fructose molecules, and this oxide linkage is formed by loss of water molecules. So when we lose water molecules, there is a linkage that is formed between glucose and fructose, and that linkage is known as glycosidic linkage. So the linkage or bond between two monosaccharide units through oxygen atom is called glycosidic linkage. Okay, now I'll explain that glycosidic linkage to you. Now in disaccharides, if the reducing groups of monosaccharides, that is aldehydic or ketonic groups, are bonded, there are these are non-reducing sugars. Example, sucrose. So in a disaccharide, okay, if reducing groups of monosaccharide, now what are the reducing groups of monosaccharide? They are aldehydes and ketones, okay, which can be reduced to carboxylic acid at any point of reaction. Now if these two groups are bonded together, okay, they are called as non-reducing sugars. Okay, an example is a sucrose. Now on the other hand, sugars in which these functional groups are free, okay, they are called reducing sugars. If these two reducing groups are bonded, then there won't be any of the things that can be reduced, because they are already bonded and are stable, so you can't reduce them. That's why they are non-reducing. If either aldehydic or the ketonic group, if either of them are free, that means you can easily reduce them. Hence those molecules are known as reducing sugars. An example is a maltose and lactose. Okay, now let's first see the structure of sucrose. Okay, the sucrose is one of the common disaccharide. Okay, so one of the common disaccharide is sucrose, which on hydrolysis gives equimolar mixture of D plus glucose and D minus fructose. Equimolar mixture means both will be in equal quantity. Okay, so when we hydrolyze sucrose, we will get equal amount of glucose. If I am hydrolyzing 10 moles of sucrose, then I will get 5 moles of glucose and 5 moles of fructose. Okay, that is equimolar mixture. Now these two monosaccharides are held together by a glycosidic linkage between C1 of alpha glucose and C2 of beta fructose. Now why C1 of alpha glucose and C2 of beta fructose? Since I told you ki sucrose is a non-reducing sugar. What does mean non-reducing sugar? That is aldehydic or ketonic group are bonded. So for glucose, carbon 1 contains the aldehydic group, and for fructose, carbon 2 contains the ketonic group. Since the reducing groups of glucose and fructose are involved in glycosidic bond formation, sucrose is a non-reducing sugar. Now you got why it is a non-reducing sugar. So here you can see both of them. This linkage is known as glycosidic linkage because glucose and fructose are joined to each other with this oxygen. So this is the glycosidic linkage, and where they are attached with the C1 and C2 of respective molecules. Glucose on C1 we have aldehyde group, fructose on C2 we have a ketonic group. Okay, so both the functional groups are the ones which are involved in bond formation, hence they are non-reducing sugar. Okay, now sucrose is a dextrorotatory, but after hydrolysis gives dextrorotatory glucose and levorotatory fructose. Sucrose in overall it is dextrorotatory, but after hydrolysis the two products, that is glucose and fructose, amongst them glucose is dextrorotatory but fructose is a levorotatory. Since the levorotatory of fructose is more than dextrorotation of glucose, the mixture is levorotatory. Now, since sucrose is dextrorotatory, but it gives a product of dextro and levo together, and the levorotation of fructose is more than dextrorotation of glucose, the entire solution is levorotatory because everywhere majority wins, right? Now, thus, hydrolysis of sucrose brings about a change in the sign of rotation from dextro, that is positive, to levo, that is negative, and the product is named as invert sugar. Now, why it is named as invert sugar is because since sucrose is dextrorotatory, but it gives a product of dextro and levo together, and since levo is present in majority, the entire mixture is levorotatory, right? Now, this brings this this brings the change in sign of rotation because initially it was positive, then later it became negative. This change in sign rotation indicates this change in sign rotation. Because of that, sucrose is named as invert sugar. Invert sugar means it is inverted from positive to negative, so it is a invert sugar. Now, second is maltose. Okay, now second one is maltose. Now, another disaccharide is maltose. Maltose is composed of 2 alpha D glucose units. Now, sucrose is made up of glucose and fructose, that is different monosaccharides. Now, here maltose is made up of same monosaccharides, 2 units of same monosaccharides. So, here maltose is made up of 2 alpha D glucose molecules, and in which C1 of one glucose is linked to C4 of another glucose. Okay, so here sucrose, both are joined with C1 of glucose and C2 of fructose. Both are functional grouped carbons, right? C1 of glucose has aldehyde and C2 of fructose has ketonic group in them. Now, for that's why it is non-reducing. As of for maltose, there are 2 glucose units which are joined together, right? Now, which carbons are the ones which are involved in the glycosidic bond formation here? Now, from one glucose, it is the C1 group, that is the aldehydic group, right? For the second glucose molecule, it is the C4 linkage. Okay, it is the C4 linkage. Now, the free aldehydic group can be produced at C1 of second glucose in solution, and it shows reducing properties, so it is a reducing sugar. Now, you got why maltose is a reducing sugar? Because if you want to make maltose a non-reducing sugar, from both the glucose molecules, C1 carbon should be the ones which would have been involved in bond formations. If both the C1 carbons were involved in bond formation, this would be a non-reducing sugar. But here we can see that for one glucose molecule it is C1 carbon, and for other glucose molecule it is C4 carbon. So this one can be converted into an aldehydic group, which can be led to reducing, okay, which can show you reducing properties. Hence, maltose is considered to be a reducing sugar. So this is the structure of maltose. Okay, and this is the glycosidic bond. Next, the last disaccharide example is lactose. Now, it is more commonly known as milk sugar. Now, lactose, it is present in milk and it is most commonly known as milk sugar. Since this disaccharide is found in milk, as I told you, since it is found in milk. Now, it is composed of beta D galactose and beta D glucose. Okay, so this lactose, it is made up of beta D galactose and beta D glucose. Now, sucrose was made up of glucose and fructose. Maltose was made up of 2 units of glucose. Now, what is lactose made up of? Lactose is made up of beta D galactose and beta D glucose. Now, the linkage is between C1 and C4 of glucose. Okay. So this is galactose structure, which is similar to glucose only. Okay, and this is the glucose, beta D glucose. Now here we can see again same thing will happen. C1 and C4. So the C1 molecule, this can get oxidized. Okay, and will show you reducing properties. Hence, lactose is also a reducing sugar. So this structure you have to remember. Okay, of both, of all the three, that is sucrose, maltose and lactose. Now, next is the polysaccharide. So we have seen monosaccharides, example glucose and fructose. Disaccharides, example sucrose, maltose and lactose. Now third one is the polysaccharides. Now polysaccharides contain large number of monosaccharide units joined together by glycosidic linkage. Now you know what is a glycosidic linkage? It is the linkage between two monosaccharides. Now polysaccharide means it contains large number of monosaccharides which are joined together by a glycosidic linkage. Now these are the most commonly encountered carbohydrates in nature. Now they are found most commonly or most common carbohydrates in the nature. Now they mainly act as the food storage or structural materials. Now what is their purpose? Okay, they mainly act as a food storage. Okay, or these structural materials. First one is the starch. Starch, you know, it's a storage polysaccharides. It is a food material of plant which is stored. So starch is the main storage polysaccharides in plants. Okay, as I told you, it is the main storage polysaccharides of plants. Now it is the most important dietary source for humans. Dietary source means we most commonly use it in our diets for our food materials. Now, high content of starch is found in cereals, roots, tubers and some vegetables. Now, we know we eat cereals. Cereals means all that moong, dal, chana and all those things are known as cereals. So, we eat lots of cereals, right? So, cereals also contain quite a large amount of starch. Again, roots. Okay, roots means potatoes, carrots. Those are considered to be roots, right? Now, potato is a basic example of a root. Potato contains lot of starch in it. Then tuber and some vegetables. Okay. Now, it is a polymer of alpha glucose and consists of two components. Now starch, it is made up of alpha glucose and it contains two components. Okay, amylose and amylopectin. Okay, it contains amylose and amylopectin. Now amylose is water soluble component which constitutes about 15 to 20% of starch. Okay, now amylose is a water soluble component and which contains 15 to 20% of starch. So in a starch 15 to 20% is amylose, which is a water soluble component, and chemically amylose is a long unbranched chain with 200 to 1000 alpha D glucose units held together by C1C4 glycosidic linkage. Now amylose, okay, it is a water soluble component which constitute 15 to 20% of starch. Okay, 15 to 20% of starch is made up of amylose, which is a water soluble compound, and chemically amylose, it is made up of 200 to 1000 units of alpha D glucose. Okay, and this 200 to 1000 units of alpha D glucose are held together by C1C4 glycosidic linkage. This is amylose. Now second one is the amylopectin, okay, which is insoluble in water, okay, and constitute of 80 to 85% of starch. So 80 to 85% of starch is made up of amylopectin. Now it is a branched chain polymer of alpha D glucose units in which chain is formed with C1C4 glycosidic linkage, whereas branching is formed with C1C6 glycosidic linkage. Now amylopectin, it is water insoluble and 80 to 85% of starch is made up of amylopectin. Now this amylopectin is a branched chain polymer of alpha D glucose. So it has a straight chain, also it has a branched chain. Now the straight chain is made up of C1C4 glycosidic linkage, whereas the branching occurs by C1C6 glycosidic linkage. Okay. Now this is the structure of amylose, and this is the structure of amylopectin. Now here you can see straight chain, it is with C1C4, whereas this branching, it happens with C1 and C6. Okay, so branch at C6 and alpha linkage at C1C4. Okay, this is the structure of amylopectin. Now next is the cellulose. Okay, now cellulose occurs exclusively in plants and it is the most abundant organic substance in plant kingdom. Now cellulose, it only occurs in plants. Okay, so it occurs only in plants and it is most abundant in organic substances in the plant kingdom. So it is found in almost most of the plants. Okay, now it is a predominant constituent of cell wall of plant cells. Now where does this cellulose is found? So cellulose is a predominant constituent. So it is the predominant constituent of cell wall of plants. So it is present on the cell wall of plant cells. Now cellulose is a straight chain polysaccharide composed only of beta D glucose units, which are joined by a glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. So cellulose, it is a straight chain structure. So you can see this is cellulose, which is a straight chain structure. Now this straight chain structure of cellulose is of beta D glucose. So all these structures are of beta D glucose. Okay, and they are joined by glycosidic linkage between C1 of one carbon. Okay, this is C1 and this is C4. Okay, so it is joined with C1 of one carbon and C1 of one unit and C4 of another unit of alpha D glucose. So like this is the structure of cellulose. So this is the structure of cellulose. Now next, third one is the glycogen. Okay, now glycogen, the carbohydrates are stored in animal body as glycogen. Now glycogen is the carbohydrates or storage form of carbohydrates in animal body. Now it is also known as animal starch. Okay, because its structure is similar to amylopectin and is rather more highly branched. So here structure is not given for glycogen, but it is observed that the structure of glycogen is similar to amylopectin. Amylopectin is a straight chain with branching, right? So the structure of glycogen is quite similar to amylopectin, but glycogen has more branches in it, okay, compared to amylopectin. So it is present in liver, muscles and brain. So where does glycogen present? Glycogen is present in liver, muscles and brain. Now when the body needs glucose, enzymes break the glycogen down to glucose. So whenever we require glucose, now glucose is important for our body, okay, because during glucose we get ATP, okay, during glucose synthesis we get ATP, and that ATP is the one which provides energy to our body, okay. This formations of ATP and this glucose structures, that things are there for you in higher classes, okay. So when the body needs glucose, enzymes break the glycogen, okay. So glucose usually we take it through our diet, so but when our diet is not proper and our body requires glucose, what happens is the glycogens which are stored in liver, muscles and brain, either one of them will start to break down, okay, to glucose, okay. Now glycogen is also found in yeast and fungi also, so it is also found in yeast and fungi. Now here the important thing is the structures, okay, structure of cellulose, structure of amylopectin, amylose, lactose, maltose, sucrose and glucose and fructose also. All these structures are important as you can get them for your exams, okay. Now next is the importance of carbohydrates. Now carbohydrates are essential for life in both plants and animals. Now till now whatever we have learned, in that we have seen ki all this monosaccharides, disaccharides, oligosaccharides, that is glucose, fructose, lactose, maltose, sucrose, starch, cellulose, glycogen, these are all present in plants and animals. So that indicates the importance of carbohydrates, right? Carbohydrates are essential for life in both plants as well as in animals. Now they form a major portion of our food. You know, carbohydrates, they form a major portion of our food. Now, honey has been used for a long time as an instant source of energy by Veds in Ayurvedic system of medicine. So many medicines, Ayurvedic medicines, they use honey, right? They mix all their tablets or those powders with honey, right? So honey has been used for a long time, okay, as an instant source of energy by Veds in Ayurvedic system, okay. Now carbohydrate, that is also a carbohydrate, okay. Now carbohydrates are used as storage molecules. We have seen some storage molecules, like for example in plants it is starch, and for example in animals it is glycogen, right? So carbohydrates used as storage molecules, example as starch in plants and glycogen in animals. Now cell wall of bacteria and plants is made up of cellulose. So cellulose is also used in plants as it is used in the cell walls of bacteria and plants. Now we build furnitures etc. from cellulose in the form of wood and clothe ourselves with cellulose in form of cotton fibers. So we build furnitures, right? All our furnitures, they are made up of cellulose from the wood, and the cloth which we wear ourselves, they those are made up of cellulose in the form of cotton fiber. Now they provide raw materials for many important industries, okay. So they became raw materials for many important industries like textiles, paper industries, lacquerers, breweries etc. Now 2-aldopentoses with D-ribose and deoxyribose, okay, are present in nucleic acid. Carbohydrates are found in biosystem in combination with many proteins and lipids. Now, 2-aldopentoses, okay, that is D-ribose and deoxyribose. So these are present in our DNA. In biology, I think you must have heard these terms, that is DNA and RNA. So it is deoxyribose and RNA is ribose nucleic acid, deoxyribose nucleic acid. So carbohydrates are also involved in biosystems in combination with many proteins and lipids. So here we have done with carbohydrates. So in our next video we'll see about the proteins, okay. So thank you for watching my video. Please do like, share and subscribe to our channel.